So good afternoon, everyone. Welcome to Cell Talk presentation session. So for today's Cell Talk presentation, Chaya is the presenter and the topic she selected is nonlinear dynamics of multi-omics profiles during human aging. Before starting the presentation, I like to introduce Chaya. Chaya is working as a data analytics intern at Prenix Life Sciences. Currently, she is collecting the details of the professors who have research interest in healthcare and sponsored projects related to standalone universities, standalone research institutes, and engineering colleges across India statewide. She completed her MTech in computer science from SVCE VTU University, Belgium in 2015. Before joining Renix, she completed an internship at the Procurement and Logistics Department of Invicta Consumer Private Limited. At Renix, she has gained experience in data retrieval and managing shared responsibilities among team members. So I welcome Chaya to start her presentation.
Hello everyone. Today my topic is nonlinear dynamics of multi-omics profiles during human aging. Um, this topic is selected on omics and longevity. The year of publication is 2024 and this research has a direct impact on aging. So coming to introduction, aging is a natural process that brings changes in our bodies, increasing the chances of developing diseases like heart problems, diabetes, and Alzheimer's and cancer. To help people to live healthier lives for longer, scientists need to understand what happens in our bodies as we age and identify ways to prevent and treat the aging related problems. While many studies have focused on changes that happen steadily over time, aging related diseases often appear suddenly after a certain age, showing that aging is not always a simple and steady process. So, coming to general background, as we age, many molecules of our bodies, like proteins, RNA, and chemicals, change and affect our body work. Scientists now have tools to study these changes in great details thanks to advanced technology called omics. Aging related health issues often increase sharply at certain ages like 40s and 60s. These sudden changes are not fully understood. So, coming to present challenges, research on aging mostly overlooks sudden changes, lacks a combined study of all biological factors, rarely tracks individuals long term, and still does not fully understand why sudden health declines and disease risk occurs. So objectives of research to find the unique changes at different stages of life, to understand why some diseases increase quickly at certain ages, and to use these findings to help to create better ways to prevent or treat age-related diseases. And methods. The study began by recruiting 108 healthy participants who gave their informed consent, meaning they understood the purpose of the research and agreed to take part. To ensure accurate results, participants with certain health conditions like anemia, kidney disease, or recent surgeries were excluded, and only those in good health were included. Each participant underwent a glucose tolerance test to measure how their body processed glucose, and based on this, they were divided into two groups: insulin sensitive individuals, those who process the glucose normally, and insulin resistant individuals, those who hold the difficulty in processing the glucose. In addition to the glucose test, other health information was also collected, including fasting glucose levels and also hemoglobin A1C to check for signs of diabetes. Um, Over time, participants returned for follow-up visits, providing more healthy samples of blood, stool, nozzle, and skin or oral. But only data from those who remained healthy were used in this study. The research examined several types of biological data to understand how the body changes with age, including gene activity, that is transcriptomics, protein levels, proteomics, and metabolites, immunomarkers, liver function, lipids, and also gut bacteria. So, all these samples were carefully processed in the lab, and advanced methods were used to ensure the results were accurate. To enhance the reliability, statistical adjustments were made to account for factors like body mass index, sex, and ethnicity. The study then tracked how these biological factors changed as participants aged, offering insights to the aging process. The main goal was to identify specific molecules that became altered with age. This research provides valuable information that could help to improve our understanding of aging related diseases like diabetes and heart diseases. So, coming to the result, the study examines the molecular changes that occur during aging using a multi-omics approach. The study involved 108 participants aged between 25 to 75 years with a median age of 55.7 years. Demographic data such as age, BMI, and gender and insulin resistance and ethnicity were collected, and you can see it on this heat map. So, broad range of omics data were collected from the participants, like transcriptomics, proteomics, metabolomics, microbes, etc. All these were collected. These data were categorized and displayed using different colors, showing how demographic factors influence the types of omics data collected. And in the figure B, you can see the data was gathered from the biological samples provided by the participants, such as plasma, stool, skin, oral, and nostril swabs. The samples were analyzed for various omics data like transcriptomics, proteomics, etc. This is the flow chart. Illustrates how these samples were collected and processed over time, emphasizing the points where data collection began and ended for each omics type. And coming to the figure C, the sample collection period varied greatly from 0 to 2000 days. Some participants were monitored for several years, while others were observed for shorter period. So red markers on the graph highlights important events or milestones in the data collection process, providing insights into the timeline of sample collection. And figure D, that is the volcano plot, visualizes statistical significance of changes in different omics categories. The vertical axis represents the significance of changes, and horizontal axis shows the degree of association with aging. This plot reveals that certain data types such as microbes and clinical lab tests exhibit significant changes as individuals age with varying levels of correlation. And this figure E. This panel provides a summary of how various molecular features changes at different ages. So colored boxes indicate significantly changed molecules, while gray blocks indicates molecules that didn't show significant changes with age. This helps identify which molecular types are more likely to undergo changes with aging. And coming to the figure F, this diagram compares the proportion of molecules that undergo linear versus non-linear changes during aging. So 81% of molecules in the study underwent the non-linear changes, while only 16.2% showed the linear changes. This data highlights that non-linear changes are more prevalent, suggesting that the aging process is more complex and less predictable than previously thought. And coming to the figure G, this is the heat map, shows how molecular levels changes across different ages, allowing the visualization of non-linear changes in molecular and microbes over time. The heat map provides a quick glance at how molecular features either increase or decrease as participants age. The visual representation confirms that nonlinear changes are widespread across different omics data types. Coming to this one, the researchers found strong links between certain types of biological data like metabolites, cytokines, and oral microbiome and the participant age. Okay, coming to the figure, we can see these are the scatter plots, shows how different groups of molecules changes with age. The correlation number tells whether the molecules increase or decrease as people age. So molecules like metabolics and oral microbiome have stronger changes with age, while the clinical lab tests have changed very little. They use the method called as partial least square segregation to compare how strongly each data type was related to age, confirming the usefulness of these data sets for studying aging. However, the data was not perfectly aligned across all the samples, so the researchers smoothed it out using a special method to make comparisons easier. Then they grouped the molecules based on similar patterns of change over time, identifying 11 groups that change during the aging. This colorful chart shows patterns of how all the molecules behave across different ages. The colors represent whether the molecules are at higher or lower levels of certain ages. Clusters with similar patterns are shown in the right. The researchers, three groups had clear patterns, that is cluster 2 and cluster 5. They showed the fluctuations before age 60, then sharply increased at age 55 to 60. And cluster 4 stayed stable until around age 60 and dropped sharply. These patterns are mostly found in gene activity data. The researchers also looked at individual participants but found it difficult to identify clear patterns in a short time span, meaning more long-term tracking is needed to observe these changes in individual level. And they also check if menopause in women could explain the transition seen around 55 years of age, but found that both men and women showed similar patterns, suggesting this change is a common feature in aging. And this figure shows how body changes as we age by looking at three types of data. Different groups of genes change in specific ways as we get older. Some genes related to cell regulation and protein production increase over time. And while other related to energy production, oxygen use, and stress protection decrease after around 60 years. Another group of genes involved in RNA processing and blood cell function increases sharply in older age. And you can see in the second one, important fatty acids and other molecules linked to the energy production and anti-inflammatory process decrease as we age. This suggests our body ability to handle fats and maintain energy declines over time. And you can see the lab test shows clear aging patterns. For example, blood urea nitrogen and glucose level, which can point, this blood urea nitrogen and the glucose level increases, which can point to the kidney issues and higher diabetes risk. At the same time, red blood cells health becomes more irregular, increasing the risk of conditions called anemia. And in this picture, we can scientists found two major peaks or waves in molecular changes at around 44 years and the 60 years of age. This peak shows significant changes in molecules and microbes in our body. The peak stayed the same even when they used different ways of test, like changing the data cutoffs and using sliding windows. When they mixed up the ages in the data, a test to check the randomness, the peaks disappeared. This means changes are truly related to the age. And these waves are like milestones in aging, marking times when the body undergoes big changes. Multiple types of biological data, like transcriptomics, proteomics, showed this same pattern, proving it's a systematic change, not just one part of the body. And the last one, this part explains how different biological functions are linked to aging peaks. Like crest 1 and the crest 2 are highlighted in two sections. The top section, pathways are linked to the crest 1, that is approximately 44 years. And bottom section, pathways are linked to crest 2, that is 60 years. So orange boxes are the function, the green boxes are the functional modules, and the blue boxes are the pathways. Lines show how these functions or pathways and processes are connected to specific genes, proteins, or metabolites. The number of connections is indicated by the size of the dots. And the pathways in the first peak, that is crest 1, involve skin muscle and alcohol lipid metabolism. And pathways in the second peak include immune functions, carbohydrate metabolism, and kidney functions. Same pathways overlap between two peaks. And this figure shows how pathways and the functions overlap between aging and peaks and clusters. The bar chart shows the number of overlapping pathways of crest 1, crest 2, and clusters 2, 4, 5. And colored bars, that is blue, orange, and black, represent specific overlaps. Dot shows pathways shared between different peaks and clusters, and horizontal lines connect overlaps between multiple peaks and clusters. And coming to the circular diagram, shows exact number of shared pathways between crest 1, crest 2, and clusters 2, 4, and 5. The red areas indicates common pathways, and their count is written inside. Okay. And the key insight suggests that while crest 1 and crest 2 share the same pathways, each also have unique pathway. And additionally, certain pathways overlap with clusters 2, 4, and 5, emphasizing their significance in the aging process that contribute to the aging-related issues like cardiovascular diseases and skin muscle aging. For instance, changes in heart health, including blood clotting, metabolism, can increase the risk of cardiovascular issues. And the overlap between two peaks indicates that similar biological processes are disturbed during both the ages. Okay. Next, coming to the discussion, the study reveals that aging does not happen gradually or in straight line, but involves complicated changes in our body molecules over time. These changes do not happen uniformly. They often slow, sharp shifts at specific ages, such as around 40 and 60. These shifts might be connected to the development of diseases, such as heart diseases and diabetes. To understand these changes better, the researchers looked at how molecules in the body behave as we change. By tracking these patterns over time, they created a clearer picture of how aging impacts the body at different stages. The study used the advanced techniques to group the molecules with similar behaviors into the patterns. This helped to identify which molecular change might be linked to common health problems, offering insights into how aging affects our overall health and risk for diseases. And coming to the limitations, the study had only 108 participants, so the result might not apply to the larger or more diverse groups. The follow-up period was short, that is 1.7 years, making it harder to capture long-term aging changes. No detailed data was collected on participants' lifestyle like exercise or diet, which can influence the aging and health. Most participants were from Stanford area, so the findings may not reflect the people from other regions or background. And molecular data was taken only from the blood samples, not from other tissues like skin or muscles. Some molecular changes may have missed if they did not follow clear patterns over time. And coming to the conclusion, the study reveals that Aging involves complex changes in the body with significant shifts occurring around 40 and 60. These changes may help explain why certain diseases become more common as people grow older. A key strength of this research is its approach of tracking individuals over time to better understand how aging unfolds. However, to confirm these findings, future studies need to be include larger and more diverse groups of participants and follow them for longer period. And impact of research. This research helps us to understand how aging affects our bodies and could help to prevent diseases like heart diseases and diabetes. By findings, by findings, important changes that happen as we age. It may lead to earlier treatments and healthier lives. It shows that aging is complex and needs to be studied in more detail. As more people around the world grow older, learning about these changes will be important to keep everyone healthy for longer. And thank you. You can find me at Chaya at renex.org. Thank you.
Thank you, Chaya, for the nice presentation. And audience, now the floor is open for any queries. So I don't see any questions. So can you move to the next slide? Yeah. Our next presentation will be on healthy lifestyles in late life, longevity genes, and life expectancy among older adults, a 20-year population-based prospective cohort study, which will be presented by Divya Lakshmi Ramakrishna on 15th Jan 2025. Thank you everyone for attending the CELTOP presentation and hope to see you all in the next presentation.